In continuation to the previous lecture, today we will start pre-treatment. At the end of a fermentation in which a microorganism rich in the required enzyme has been cultured, the broth may be cooled rapidly to 5 degrees centigrade to prevent further microbial growth and stabilize the enzyme product. The pH may also be adjusted to optimize enzyme stability. If the enzyme producing organism is a fungus, this may be removed by centrifugation at low speed. If the enzyme source is bacterial, the bacteria are often flocculated with aluminum sulfate or calcium chloride, which negate the charge on the bacterial membranes, causing them to clump and thus come out of suspension. Now the treatment. Extracellular enzymes are found in the liquid component of the pre-treatment process. However, intracellular enzymes require more extensive treatment. The biomass may be concentrated by centrifugation and washed to remove medium components. The cellular component must then be ruptured to release the enzyme content. This can be done using one of the more of the following processes. First is ball milling using glass beads. Second is enzymic removal of the cell wall. Third is freeze-thaw cycles. And the fourth is liquid sharing through a small orifice at high pressure within a French press, osmotic shock or sonification. Separation of enzymes from the resulting solution may then involve a variety of separation processes which are often employed in a sequential fashion. The first step in an enzyme purification procedure commonly involves separation of the protein from the non-protein components by a process of salting out. Proteins remains in aqueous solution because of interactions between the hydrophilic water-loving amino acids and the surrounding water molecules, the solvents. If the ionic strength of the solvent is increased by adding an agent such as ammonium sulfate, some of the water molecules will interact with the salt ions, thereby decreasing the number of water molecules available to interact with the protein. Under such conditions, when protein molecule cannot interact with the solvent, they interact with each other, coagulating and coming out of solutions in the form of a precipitate. This precipitate containing the enzyme of interest and other proteins can then be filtered or centrifuged and separated from the supernatant. Since different proteins vary in the extent to which they interact with water, it is possible to perform this process using a series of additions of ammonium sulfate, increasing the ionic strength in a stepwise fashion and removing the precipitate at each stage. Thus, such fractional precipitation is not only capable of separating protein from non-protein components, but can also enable separation of the enzyme of interest from some of the other protein components. Subsequently, a wide variety of techniques may be used for further purification, and steps involved chromatography are standard practice. Ion exchange chromatography is often effective during the early stages of the purification process. The protein solution is added to a column containing an insoluble polymer, example cellulose, that has been modified so that its ionic characteristics will determine the type of mobile ion, that is cation or anion, it attracts. Protein whose net charge is opposite to that of the ion exchange material will bind to it, whereas all other proteins will pass through the column. A subsequent change in pH or the introduction of a salt solution will alter the electrostatic forces, allowing the retained protein to be released into solution again. Gel filtration can be utilized in the later stages of a purification protocol to separate molecules on the basis of molecular size. Column containing a bead of crossed-linked gel particles such as Sephadex are used. These gel particles exclude large protein molecules while allowing the entry of smaller molecules. Separation occurs because the larger protein molecules follow a path down the column between the Cephadex particles, occupying a smaller fraction of the column volume. Larger molecules therefore have a shorter elution time and are recovered first from the gel filtration column. Affinity chromatography procedures can often enable purification protocols to be substantially simplified. Typically, with respect to enzyme purification, a column would be packed with a particulate stationary phase to which a ligand molecule such as a substrate analog, inhibitor or cofactor of the enzyme of interest would be firmly bound. As the sample mixture is passed through the column, the enzyme interacts with and binds to the immobilized ligand, being retained within the column as all of the other components of the mixture pass through the column unrewarded. Subsequently, a solution of the ligand is introduced to the column to release elute and thereby recover the bound enzyme from the column in a highly purified form. Nowadays, numerous alternative affinity chromatography procedures exist that are able to separate enzymes by binding to areas of the molecule away from their active site. Advances in molecular biology enable us to purify recombinant proteins, including enzymes, through affinity tagging. In a typical approach, the gene for the enzyme of interest would be modified to code for a further short amino acid sequence at either the nitrogen or carbon terminal. For example, a range of polyhistidine tagging procedures are available to yield protein products with 6 or more consecutive histidine residue at their nitrogen and carbon terminal end. When a mixture containing the tagged protein of the interest is subsequently passed through a column containing a nickel nitrolo triacetic acid, agarose resin, the histidine residue on the recombinant protein bind to the nickel ions attached to the support resin, retaining the protein, whilst other protein and non-protein component pass through the column. Elution of the bound protein can then be accomplished by adding imidazole to the column or by reducing the pH to 5 to 6 to displace the HIS tagged protein from the nickel ions. Such techniques are therefore capable of rapidly and highly effectively isolating an enzyme from a complex mixture in only one step and typically provide protein purities of up to 95%. If more highly purified enzyme products are required, other supplemental options are also available, including various forms of preparative electrophoresis, example disc gel electrophoresis and isoelectronic focusing.